All right. Can everybody hear me?
Yes.
All right, cool. So good morning, everybody. I'm very happy to introduce today our speaker, Vincenzo Ellis. I'm very happy to have him here. Vincenzo got his bachelor in biology at the University of California, Santa Barbara, and then he did his PhD at the University of Missouri, working with Robert Wycliffe. It's basically one of the biggest ecologists of our time. And at that time, Vincenzo did amazing work studying diversity and host specificity of avian malaria parasites. So Vincenzo did a fantastic PhD, and then he moved to Brazil, to the Federal University of Minas Gerais for a postdoc. And at this time, I met Vincenzo and we worked together. And he basically he became my unofficial PhD advisor. So he helped me out a lot, and I was so lucky to have him helping. And then was fantastic to work with Vincenzo. And then he moved to Sweden now for a second postdoc. And at this time, if I'm not wrong, Vincenzo kept working with host specificity and diversity in avian malaria parasites and other avian blood parasites. But also he started working with genomic approaches to study these parasites. So he was now developing other sets of skills, and because those skills were showing up and then getting recognized, he joined then University of Delaware as an assistant professor at the Department of Entomology and Wildlife Ecology, right? So then Vincenzo joined the University of Delaware in 2019, where now he's currently a professor and working with disease ecology and molecular ecology. So Vincenzo is a fantastic person, a very nice person, and a very successful researcher. He's done amazing work, and he's also a wizard in large-scale data analysis. He's a wizard in R, and you'll see him working in R. Is like a magic. So he's a great researcher, I guess. Very good professor, and I'm pretty sure everybody here today is gonna love his talk. And I'm very excited to have him speaking to all of us today. And that's it, Vincenzo. If you wanna say something else about your background that I didn't mention, which is maybe important for us here, go ahead. But thank you again, and thank you for taking the time to talk to us today and for accepting Dina's invitation. And hope to see you here in person soon.
Oh, thank you so much. Thank you so much, Chico. That was a wonderful introduction, and I really appreciate it. And thank you, Dina, and everyone else for inviting me and showing up for this talk. Just like Francisco was saying, I came here in 2019. The only addition that I can say, I still work with the avian malaria parasites, but my lab also has a big focus now on Borrelia in ticks, and so we've been doing a lot of work in that area as well, and looking forward to collaborating with you guys, and yeah. So that's that. I'd like to get into the talk here, and this is going to be about avian malaria parasites, as the title showed. But hang on one sec. Let's make sure I can screen one. There we go. And all right, can you see that? Looking good. All right. So host specificity of avian hemosporidian parasites. The subtitle here is how it relates to long distance dispersal. And that's really the key of the talk here. And so what I want to start with is the context for this talk, which is also the context of our lives for the past couple of years now, right? And that is emerging infectious disease. The SARS-CoV-2, the coronavirus that's made this pandemic and all these horrible things, is an example of an emerging infectious disease. And so how do we define emerging infectious disease? That's, I think, the first key thing to bring up here. And so emerging infectious diseases are parasites or pathogens that appear in new geographic areas or new host species, or even that have recently become more prevalent than they were previously. And so I want to give another example. We know about SARS-CoV-2 and switching to humans from bats and spreading rapidly across the world. This is a classic example of an emerging infectious disease. But let's look at one more that will sort of lead us into what I want to discuss in terms of dispersal. This is West Nile virus. So this is a mosquito transmitted virus that first appeared in the US, so first emerged in New York in 1999. And so where did it come from? You know, it wasn't here before. Where did it come from? And so what researchers did was they sequenced DNA, or well, they sequenced genomes of West Nile virus strains that were known, and they put them together into a phylogeny. And what they found, so this is that phylogeny from a paper published in the year that the virus emerged. And what they found was that this West Nile virus strain, I think maybe you can see my mouse here. This West Nile virus strain in the red circle here, New York 1999, is most closely related to an isolate that was found in the brain of a dead goose in Israel. So this suggests, of course, that there must have been some dispersal, a long distance dispersal across the Atlantic to get this virus to the United States. And so this is a sort of a pattern that we see of long distance dispersal, emerging disease. And we might want to ask ourselves, all right, can we predict what makes a pathogen able to become an emerging infectious disease? You know, what is it about certain pathogens that allow them to do these long distance dispersals, to change hosts? And this question has been asked a number of times, and I want to just bring our attention to one particular hypothesis, and that's shown here in this graphic. So this is a paper that looked at pathogens and parasites of humans, and then ranked them. They checked the percentage of them that were emerging, right? So maybe 30% of the group are emerging. That's on the y-axis here. And on the x-axis, they separated them by the number of host species they infected. So if they have 0, it means they just infect humans. If it has 1, they infect humans plus 1 other host species, 2, humans and at least 2 other host species, and onwards, and so on. And so the point here is that the pathogens, the parasites that are able to that are more often emerging, more likelihood of emerging, seem to be host generalists, at least in humans. So, and this kind of makes some sense, right? So the more hosts that you have, this is a measure of what we call host specificity, which is the point of this talk here. The more hosts you infect, we consider you a generalist. And if you infect 1 host or just a couple maybe closely related hosts, you are considered, the parasite is considered a specialist. And so we see that generalists are associated with a higher percentage of emerging parasites and pathogens in humans. And this makes sense, right? A generalist parasite that can jump to a new area, or that, by chance finds itself in a new location, maybe it's more likely to be able to infect the hosts that it encounters than a specialist would. A specialist infects 1 host species, you put it in a new area with new hosts around, and maybe it can't get in. So this makes some sense. But to really get at this hypothesis of long distance dispersal leading to emerging infectious disease in relation to, as a result of host specificity, one would like to do more than compare viruses and helminths or different kinds of viruses. What's better is if we can look within a group, a clade of related parasites and pathogens. And where we have, variation in host specificity and evidence of dispersal. And what I would like to argue is that avian hemosporidians are an excellent model for testing this hypothesis of whether long distance intercontinental dispersal, that's what we're really going to be concerned with here, is related to host specificity. So whether generalists can disperse these long distances across continents more readily than specialists. And so, yeah, what are avian hemosporidians, right? So avian hemosporidians, we often call them malaria parasites or avian malaria parasites. And this is a relatively recent phylogeny of malaria parasites, of hemosporidians. They're apicomplexan parasites. The order is Hemosporida. And you know some of them. I don't know if this is perhaps a little too small of the writing here, but here's Plasmodium falciparum in the middle of the red clade, if you can see my cursor. Major pathogen of humans, kills lots and lots of people every year. Here's Plasmodium vivax, another human at the top of the phylogeny, if you can't see my cursor, another human malaria parasite. But the rest
Vince,
of the...
Vince, sorry, I'm going to
Yeah.
have to do this, and I apologize, but we're having problems with, we're not being able to see your slides 100%. We see your cursor, by the way, not a problem, but for some reason, the view that you're using, it gives us a tiny little slide, a little, we can't do a full screen on our screen. So just pointing out,
right?
Sorry?
Yeah, the view is like vertical view from his screen instead of being horizontal.
Oh, good point. Maybe that's what it is. If so, we were having some conversation on the chat. I really didn't want to
sorry,
interrupt you.
no problem, no problem.
But once you started going on a slide like this, I'm like,
Yeah, hang on.
You don't mind.
No
Yeah,
problem.
I think Francisco, that's the exact right thing. It's a shape thing. If it doesn't work, we can totally see it, and I'm just very close to my screen. That's the only thing.
Yeah. Let's hold on. Okay.
I'm sorry.
No. Let's see if I can do it like this. If I just disconnect the other monitor and then. Sorry about that, guys.
Okay.
Let's go. Let's just share screen now again. Okay, so now you can see my.
Yes.
Okay, all right.
There we go.
Okay, and how about now?
Yes, that's better.
Okay, much better. Okay, sorry about that. Yeah, my apologies. Yeah, new don't usually zoom from this location. So all right, so yeah, okay. So the point I want to make here though is that avian hemosporidians are and hemosporidian parasites are part of this large clade that includes human parasites, but also parasites of birds. As you can see, the hosts are plotted here on the right side of the phylogeny. And so within this context, you have bird malaria parasites, you have human malaria parasites, you have parasites of bats and monkeys and mice and things like that. What I'm concerned with here are these groups, the Plasmodium bird parasites. And so these are images of bird red blood cells and a parasite in the middle here of all of these. So these are Plasmodium parasites. In the middle here, we have Hemoproteus parasites. These are transmitted by biting midges predominantly, at least among the ones that infect songbirds. And these are leukocytozoan parasites that are transmitted by black flies. And in an entomology department, you probably know all about black flies. My least favorite of these vectors here. I just don't like getting bit by black flies. But anyway, these are our parasites that make up what we call the avian malaria parasites. And so, how do they? What's the life cycle of these guys? The important thing here, they reproduce sexually in their dipteran vector. The gametes combine, they form a new stage that goes to the salivary glands. The vector bites a vertebrate host. Here we're talking about the avian malaria parasite, so a bird. And then the parasite reproduces asexually in the solid tissues or in the blood or both, and then get picked up again by a mosquito vector. And often people ask me, when I talk to them about avian malaria, but so where does the vector bite the bird? Birds are covered with feathers. How is that possible, right? And so here's just an example I like to show. At least some image that I've seen it happen. This is a Eurasian blackbird being pulled out of a mist net, and as soon as I pulled it out of the net, 2 mosquitoes just boom come down as soon as right where the feathers had parted. So anywhere that they can bite is where they will bite. And so what we do typically for surveying for these kinds of parasites, we're catching birds or perhaps catching vectors. For the birds, we take a small blood sample. We can make a blood smear that we stain. That's this image up here. And we also take blood for genetic analysis. And with the smear, we can morphologically identify parasites. So here you can see a Hemoproteus parasite in a bird red blood cell. These dark centers of the cells is the nucleus of the cell. Birds have nucleated red blood cells. And here on the bottom, we have just a small, short bit of sequence. The avian malaria community is sort of settled on a barcoding scheme. And so we can define lineages of avian malaria parasites, which often, not always, but often can seem to correspond to good species. Based on about half of their mitochondrial cytochrome B gene. And importantly, these records for avian malaria parasites are kept in a central location. This is the Malawi database. It has a global, publicly available, open access type of database. Run by Staffen Bench. This is a picture of Staffen with a buzzard in his hands here. As you can see, this is also probably the best picture I've ever taken in my life. And so Staffen was my postdoc advisor in Sweden, and Staffen runs and maintains and curates the Malawi database. Importantly, it has information on the hosts that lineages infect and the locations that they're found in globally. And so just to show you a little bit here, when we look at the number of host species that these parasites infect, and that's shown in these frequency distributions here, we find that a lot of the parasites are often found one time or found on just one host, so very specialist or perhaps not sampled very well. But we see a lot of variation, actually. We can see some in multiple hosts and even some really extreme generalists in some of the parasite genera. We also see this sort of distribution that's global, right? They're found in all the continents except Antarctica, these parasites. So this is a map showing countries highlighting where the number of studies that have been deposited on Malawi that have found parasites. And so this is a little bit old now, so there's more countries that could be filled out here. But again, globally distributed except in Antarctica. And so I want to take a quick stop here and do a little quiz and ask you, what type of zoo birds do you think get sickest from avian malaria infections? And now I haven't been looking at the chat, but I'm gonna go into the chat here. All right, yeah, what type of zoo birds do you think get sickest from avian malaria infections? And I've given you a hint already, so. But you can just guess, yeah. You can unmute or you could just throw it in the chat. What kind of birds do you think get sickest from avian malaria? Oh, I see a correct answer right straight away. Yes, it is absolutely it's absolutely penguins. Yeah, it's penguins. And this is right. Yeah, nice work. And this is, because as I said, they're not in Antarctica. This is immunologically naive hosts typically. Okay, so moving along. No more quizzes in the rest of the talk, don't worry. Okay, avian hemosporidians as emerging infectious diseases. So avian hemosporidians, the very famous example is the avian malaria parasite in Hawaii. So in around 1830, this mosquito, Culex quinquefasciatus, was introduced to the Hawaiian Islands. And this allowed for the introduction and establishment of an avian malaria parasite, Plasmodium relictum, about 100 years later, probably from the release of caged birds. And so here's of course, the Hawaiian Islands. This is an image of a Plasmodium relictum, a parasite, and the Culex mosquito here. And this parasite has been terrible for native Hawaiian birds, right? And so this is a classic study from Van Riper and colleagues in 1986, and it's showing that that vectors are typical. So this is elevation on the x-axis, and what we see is that Native birds are seeming to be restricted to high elevations because of malaria parasites. So the malaria parasites are able to be transmitted by vectors at the lower elevations and have really limited the distribution of native birds. So it's really been a devastating emerging infectious disease of wildlife in Hawaii. So avian malaria is these emerging infectious diseases. It turns out that the cytochrome B lineage, remember I told you that we can sequence a short fragment of the mitochondrial gene to get lineages of these parasites. The lineage for the parasite that's in Hawaii is known as GRW4. And it's been found all over the place. It's been found in 43 countries, 84 host species, mostly passerines, but 5 bird orders in total. It's definitely an extreme disperser and extreme generalist. You know, whether many of these introductions or dispersal events are human mediated or not, it's certainly gotten to a lot of places and infects a lot of host species. So it's sort of another fitting into our hypothesis of host specificity, generalists doing well at dispersing and establishing in new areas, becoming emerging infectious diseases. And in fact, when you dig into the Malawi data set and the avian malaria data set, you can actually see that that many lineages are in fact found in more than one biogeographic region. They're found distributed across the globe, suggesting successful dispersal events. So here what I've done is just broken up the world into some biogeographic regions here, and I've counted the number of regions occupied by each lineage in the Malawi database. And so most lineages, of course, are found in one area, for instance, North America or Sub-Saharan Africa or South America or something. But there's actually quite a few that are found in more than one biogeographic region, so big areas, suggesting some dispersal here. And so one can ask, you know, is there a correlation between host specificity and the number of continents, for instance, that a lineage is found on? So here I'm switching to quantifying the number of continents a lineage is found on rather than those biogeographic regions. So this is the same graphic now showing us the distribution, the frequency distribution of the number of continents that lineages are found on. So again, most lineages of Plasmodium, Hemoproteus, and Leucocytozoan are found on one continent, but there are many lineages that are found on more than one continent, suggesting long distance dispersal. So is that related to host specificity? That's what I want to ask here. And if we do a sort of simple correlation between the number of host species that a parasite infects and the number of continents that the parasite occupies, we can see the positive correlations. They're weak, of course, but they are positive correlations that we would expect. So the more continents occupied, the more hosts the parasite infects. You also might expect that, right? Because it's not the same host species on multiple continents. So if a parasite's going to get to a new continent, they're often going to be infecting new species anyway. So you expect this relationship. We can also turn this around and look at, well, not turn it around, but just look at it from a different lens. Instead of looking at number of host species that the parasites infect, we can look at the phylogenetic host diversity here. And I think this is mean pairwise phylogenetic distance of the hosts that a parasite infects. But again, similar thing. We see some significant and perhaps weak positive correlations as we would expect. So the more continents, so each of these points here is a parasite lineage. The more continents that the parasite occupies in general, the more hosts and the more phylogenetically diverse hosts it's going to infect. And so, okay, so, but let's take, let's remember back to that example of the West Nile virus, right? And we looked at that West Nile virus. There was a lineage. The first one to appear in New York was most closely related to one from a goose in Israel, right? And we inferred a dispersal event. So let's just take a quick look at how we can infer dispersal events from phylogenies. I just want to show this just a quick demonstration here. Imagine this, this is a fake phylogeny. We have species 1, species 2, and species 3. Species 1 is distributed in region A, species 2 also in region A, and species 3 is in region B. So we see species 2 and 3 are sister to each other, and then this clade is we could say is sister to species 1 here. So what can we infer about dispersal events by looking at that phylogeny? Well, we can say, we can go back to the nodes, right? The common ancestors here of the species here. And we can say, well, the most parsimonious result here is that there was a local diversification here at this node that led to species 1 in region A, that led to species 1 and whatever the ancestor of species 2 and species 3 was, this node. That was a sympatric speciation event. The speciation event happened in region A. Whereas here we might infer that that there was an allopatric, a dispersal event that led to species 3 becoming distributed in region B, right? So there's a speciation event that leads to species 3 dispersing to region B. So perhaps some individuals dispersed out to region B, and then over time they become a new species. So we can classify, we can categorize nodes as sympatric or allopatric that tell us something about whether there was dispersal or not. And one can do this for avian hemosporidian parasites, for avian malaria parasites. The problem is that for most of these parasites, we just have that about 500 base pairs of barcoding region to go on. And when you try to build a phylogeny, there's thousands of these parasites. There's probably, there's probably 4,000 or so lineages identified, and probably we'll get to, 10,000 or so, is the same number of bird species. When you try to build a phylogeny with that small amount of genetic information, they're often not well supported. So this is this is probably the Plasmodium phylogeny, I think. And what I've done is just colored nodes red with a higher bootstrap support in a maximum likelihood type of analysis. And so we can kind of focus in on those red highly supported nodes to look at what we can infer about dispersal. And when you do that, and you just start classifying these well-supported nodes as whether they're allopatric or sympatric, that is, whether there seems to have been a diversification event within a certain area or across two different areas, right? So whether there was dispersal would be allopatric. We see actually quite a big fraction of those nodes being allopatric dispersal events. So, 11% of the Plasmodium nodes that I've identified here are on one side of the node, you have one region, and another side of the node, you have another region. 16% for Hemoproteus and 15% for Leukocytosome. So again, suggesting that there has been this intercontinental dispersal that leads to speciation. And so, okay, where has the dispersal occurred? This is also part of the question here, because of course birds migrate, right? We know that birds migrate. They're often can be moving across continents, and so where does the dispersal occur? And of course, we do see dispersal across avian migratory routes. So what I'm showing here, if you can see these black lines here are just connecting sister lineages. Okay, so here's a sister lineage in Central America, or a lineage in Central America, and its sister in North America. Right, here's one in Europe, and its sister in Sub-Saharan Africa. These fall along avian migratory routes, kind of what you might expect for parasite dispersal in that sense. On the other hand, we find examples of sister lineages that don't follow avian migratory routes. For instance, a lineage here, this is a lineage here in North America. Its sister lineages in Sub-Saharan Africa. Here's one in North America and its sister in Europe. Here's one that's distributed in Asia and its sister in South America. So we see some examples of these really strange long distance migratory events that are not along migratory routes. How does that happen? Maybe it's people moving them in some cases. That certainly was the case with the Hawaiian example, but in other cases, maybe birds get blown off course, maybe mosquitoes or vectors get blown off course. Unknown, but clearly something is happening here. And so we can now, let's sort of dig deeper into this and say, all right, so do sister lineages on different continents infect more host species than sister lineages on the same continent? In other words, are the sisters that the sister lineages, the lineages that we know have dispersed across continents, right? So that you have two lineages, they're each other's closest relative, and they're on two continents. That suggests that they dispersed long distance. Are they infecting more host species than the ones that have the sister lineages are found in the same continent, you know, because that would, again, going back to that hypothesis that host specificity is important in dispersal events, right? So do generalists disperse more? Can we interrogate this data set to ask that same question? And when you do that, you find that no, not really, actually. So these plots here are showing each point here is representing a pair of sister lineages. Okay, and what I've plotted on the y-axis is the number of host species of the most generalist of those two lineages. Okay, so and that's sort of I think the conservative way to go. It doesn't matter if you used average either. But so we take the two lineages, we say, all right, which one of you is the most infects the most host species? All right, I'm going to put that on the plot here, and I'm going to put on the x-axis whether that point should be allopatric, that is, the two sisters are in different continents, and so long distance dispersal, or sympatric, they're in the same continent, no long distance dispersal. And so what one might expect is if host specificity was playing a role in dispersal, that you would see that the allopatric, the ones that are on different continents, have higher, they're more generalized, they infect more host species. And in fact, you don't see that. With Plasmodium, this P of 0.05, I mean, the sympatric is actually slightly higher than the allopatric. That's the opposite of what you might predict. But it's just a, you know, it's not a significant result. There's a lot of tests here. When we look at phylogenetic host diversity, so that's the mean phylogenetic distance of the hosts, and do the same plot, it's the same deal. We don't see real differences here. So what does that mean? What it means is that specialists can disperse long distances too. That's what it means. Specialists can also disperse long distances. And so I just want to give you an example of that. This is there's a parasite called, the species of the parasite is called Hemoproteus nucleocondensis, right? But the genetic lineage is referred to as GRW1. And you know, that's kind of a weird GRW1. GRW1 refers to great reed warbler, that's this guy here. And one, because it was the first lineage that was found in great reed warblers. Okay, and so it turns out to be a common parasite of great reed warblers. Acrocephalus aeruginaceus. These are European birds that migrate to sub-Saharan Africa, but breed in Europe. And this is a specialist of these birds. Lots and lots of birds have been screened, and we find GRW1, Hemoproteus nucleocondensis, just in great reed warblers, except for one other species. It's one other place, and that is in the Seychelles warbler, Acrocephalus seychellensis. GRW1, the same cytochrome B lineage, is also in the Seychelles, this small group of islands here to the east, well, so northeast of Madagascar, you can see in the map inset here. So the Seychelles warbler are also infected by GRW1, by Hemoproteus nucleocondensis. So that's kind of strange, right? This is a specialist parasite. So what is going on here? Well, what we did was to sequence the full mitochondrial genome, right? So it's about 6,000 base pairs. Well, here we sequence 5,851 base pairs, and we find actually 12 differences. So even though they're the same in our barcoding region, we see 12 differences between those parasites across the mitochondrial genome, a 0.2% divergent. So when did they diverge, right? When did that happen? When did the parasite get to, you know, when did this happen? There's a lot of sort of controversy about how to date, you know, the molecular rate of evolution of malaria parasites. But I just took a bunch of rates that I could find in the literature and tried to calculate this 0.2% divergence. And what we know is that for sure, among all the rates, it was less than 600,000 years ago. They diverged less than 600,000 years ago. Doesn't matter what rate in the literature that you choose. Still probably happened quite some time ago, probably thousands of years ago, but no more than 600,000 years ago. So, okay, about, let's say, less than 600,000 years ago, when did the Seychelles warbler get to Seychelles, right? Well, here I've just taken, I just went in GenBank and took I think this is 5 genes for Acrocephalus warblers, and made a phylogeny here. And I've time scaled it using the cytochrome B bird cytochrome B rate of evolution here. And we can see Acrocephalus seychellensis here in this phylogeny. And the node here is the estimated time for this node, 1.76 million years ago. And then this is its sister here of the Seychelles warbler is the Madagascar, an endemic Madagascar warbler. And so those diverged from each other about 2 million years ago. And the ancestor of those two diverged from the ancestor of their other close relatives, mainland species, about 4 million years ago. So, okay, so what does that tell us? That tells us that the host colonized, so the Seychelles warbler, the host colonized Seychelles 2 to 4 million years ago. Okay, and so, all right, Seychelles warbler colonized 2.4 million years ago, or 2 to 4 million years ago. This suggests very strongly that there was a natural long distance dispersal event. The parasite got there after the Seychelles warbler was already established. Clearly, people didn't get to the Seychelles until the 1700s. And it's certainly the divergence of 0.2% must be greater than before the 1700s. This is like almost certainly a natural long distance dispersal event. And I know, how did it happen? I don't know, maybe a great reed warbler was migrating down the east coast of Africa. A storm blows in, it blows it into the Seychelles, and Seychelles happens to have 3 biting midge species that are there. Maybe one of those bit the bird, got GRW1, transmitted it to the Seychelles warbler. Maybe that's what happened. But interestingly, GRW1 is a specialist and it doesn't infect other Acrocephalus warblers despite apparent opportunities. There's many Acrocephalus warblers in Africa and have been screened, and GRW1 does not show up. It is just in great reed warbler and in Seychelles warbler. So a specialist with this long distance dispersal. So I think that's just, to me, that's just a remarkable thing here. But let's return to the parasite on in Hawaii. All right, we started with talking about that Plasmodium relictum parasite, and it's an extreme disperser, right? Very generalist. So there was a study done by Olaf Helgren, 2015, that sequenced a segment of the Merozoite surface protein 1 gene of Plasmodium relictum lineages. And here we're seeing all the different haplotypes. These are, let me move my zoom window here. These are these PR5, 6, 4, 8 around the world here of the GRW4 lineage. So we have, this is a nuclear gene that's involved in MSP1 is a nuclear gene involved in infecting host species. It likely evolves much faster than the mitochondrial DNA of the parasite. And what he showed, or what that group of authors showed, was that there's actually 2 MSP1 haplotypes in Hawaii, PR8 and PR9. And they are also found in North and South America. Interestingly, PR8 was also found in a house sparrow in New Zealand. And it's interesting that house sparrows were introduced to Hawaii in 1871 from, I think from New Zealand. That's probably right. I have to double check that, but I think that's right. And so we have these two lineages here. But the thing is that when you look at the islands of Hawaii, PR8, haplotype PR8 seems to be restricted to Kauai, right? And so that's just remarkable. This lineage, this parasite that's all over the world, that can get all over the place, infect all sorts of birds. And we have this one of the haplotypes is restricted to Kauai. And so that's just remarkable. And I wanted to see if this was, you know, are these really distinct lineage? I mean, what's the difference between PR8 and PR9? And so as part of a separate project, we developed, along with Olaf Helgren, I developed a sequence capture protocol that targets, it targets a little over 1,000 genes in the Plasmodium relictum genome. This is where you pull genes out of solution. We have to do that with the parasites. They're really difficult to sequence because the bird red blood cells have the bird genome in them. And so you can't just do sort of deep sequencing directly on DNA extracted from a bird. So sequence capture is a way to pull out the parasite sequences with, you use RNA probes to pull them out. And so this is just a phylogeny of GRW4 lineages, that's that Plasmodium relictum lineage, based on just 25 genes that I pulled from the sequence capture protocol. And this is, the bottom one here is GRW4 from PR8, and this is PR9. And so they actually come out as being quite divergent across a number of genes, not just that small section of MSP1. So it could be that this is sampling. Maybe more sampling would find PR8 down in the other islands. But if it's not, it's pretty interesting and seems to not be quite in keeping with this idea of host specificity and dispersal. So what do we make of all this? And I promise I'm wrapping up here. What do we make of all of this? I think that the point that I want to make, host specificity must be involved in dispersal of parasites and pathogens. At some level, it must be, because they have to be able to infect novel host species that they encounter for them to be successful in the dispersal event to lead to something, to lead to the establishment of the pathogen. But the patterns aren't clear, and specialists are able to disperse long distances and infect hosts in new places. And so I guess the real take-home message for me is, or from me, is that predicting emerging infectious diseases is not going to be easy. You know, this is just not some very simple, theory will predict what's going on here. And so with that, I just want to end the talk. A lot of people have been involved working with me, and I've been working with a lot of people on different things. The malavi analysis was with Staffen Bench in Lund University, and Tiajong Fan was at University of Delaware, now at UC Davis. The GRW1 Seychelles analysis with Arif Çiloğlu from Erciyes University in Turkey, and also Staffen Bench, and the GRW4 Hawaii work with Olaf Helgenen and Carter Atkinson Olaf from Lund University and Carter from USGS. So yeah, with that, thank you so much again, and I'm happy to take any questions. I'm going to stop my share here.
All right, so raise your hand if you have questions, and step right up. Again, we're not going to have time to talk to Dr. Ellis after, so this is the moment. I have a ton of questions, so please step up.
Yeah, can I ask without raising my hand first? I don't see any other hand.
Come on, go, Dana, go.
Hi, Vincenzo. Yeah, thank you very much. I'm going to save probably most of my questions for when you can visit, because I have lots of them, but I'm going to ask the most pressing one because I'm about to talk about it on Tuesday.
Okay.
I don't want to say anything stupid. So in your opinion, is what's in malavi database also reflected or gleaned from NCBI? Is it fair to say that they're homologous? GenBank specifically, the nucleotide sequences.
The naming is problematic, and so you can find things in GenBank and NCBI that you can find the same thing named different things. So I guess if you were just to extract the sequences, it would be okay. I think malavi gets stuff before it gets onto GenBank, so it's often a little bit ahead. Malavi also sometimes in GenBank, people don't put all the host species that a lineage was found from. In malavi, they typically should. So I would say malavi is much better. Malavi's also been checked, so it doesn't make it into malavi without Staffen doing a pretty thorough check on the data set. So I would say it's a kind of course, GenBank is highly curated, but malavi's curated specifically for these avian malaria parasites. So okay. Yeah.
I'm asking because I have a large metagenomic, including RNA, meta-RNA omics data sets from mosquitoes in our area. And there's a large diversity of Plasmodium in there, much more than I had thought I was going to see. But strictly ripping the small subunit RNA sequences out showed, using a curated database like Silva or something, small subunit databases showed things that weren't there. Gallinaceum turned out to be elongatum, and Voganai was there if I just looked at NCBI, and I'm wondering if I should use cytochrome B instead of the small subunit RNA.
Oh yeah. Yeah,
now.
yeah, yes, yeah.
Because what's clearly right, you can see homology 99-100% at the small subunit, which is fine, but then there are other things that look like, for example, gallinaceum that are only 92-93% homologous, and it's clearly not gallinaceum, but it's also not anything that's in NCBI.
Sure.
So I can go back and check to see if there's maybe some site B in there and look in malavi, but there's this really large diversity of plasmodiums in our mosquitoes that I right now it looks undescribed as far as NCBI goes. So I'd be curious.
The mosquitoes from where?
New Jersey, right here in our area.
Okay, cool. Yeah. I mean, I think another thing is that, I think there's a lot of diversity yet to be discovered. Almost certainly. I mean, I think malavi is probably up to around 4,000 cytochrome B lineages, but I mean, there's a lot more left. I wouldn't be surprised if there's as many lineages as there are species of birds. So
yeah, okay.
So Jim put a question in the chat. Why don't you go ahead, Jim?
Yeah, I have to ask. I'm very tick centric. Did you remove the ticks from the Eurasian blackbird?
that's great question. No, we did not.
Oh.
Yeah. Too bad, right? We not that blackbird, but we did remove several ticks from birds when we were netting in Sweden. They're sitting in a freezer somewhere, I think.
Awesome.
I don't know exactly where, but yeah, they're there. There's always yeah. No, ticks are great. I mean, now I'm working with ticks a lot too. So yeah.
Yeah, but
Thank
Jim
you.
says there's actually, you can see it in the picture. So I was looking at the mosquitoes. I totally missed the ticks. So it's actually on the picture that you showed.
Okay, I'll look back closely at that. That's great.
So
Yeah.
Okay, so since nobody else is like lifting their hand, I'm gonna sort of, because I'm gonna go sort of to the basis of your thesis. Because there's a couple of things. There's invasive birds, right?
Sure,
So
yeah.
you don't need to have a different host when you move to a different continent. You can simply go feed on the European starlings or the European sparrows that are everywhere, right? So
Yes.
how does that? So that's like question on how does that mess up your set of assumptions to do these and then it has to do sort of this timeline, but you kind of addressed some of that about what is a species. I mean, you had a 0.12% difference in GRW1 between the Seychelles and you're still calling that, I'm assuming, the same species. In fact, how?
Yeah, that's great. Both points. Yeah,
Okay.
both points really interesting. So the question about whether they, some of the dispersal patterns that you see of the parasites, I would not be surprised if many of them had to do with invasive species, with things like house sparrows that Plasmodium can infect. For others, probably, I don't think it does. I mean, I think it depends on the. I don't know if that represents sort of explains the vast majority of the variation in the dispersal. It probably explains a significant chunk of it. On the other hand, you have these parasites that are infecting really kind of phylogenetically different host species in different areas. And so, yeah, it could be that maybe there's a reservoir of an invasive species that we just haven't sampled yet. But on the other hand, I think that there's probably real sort of not well understood biological phenomena going on here of parasites being able to just get into new host species. I don't know how it works.
But I mean, does it take a special kind of parasite to infect an invasive species like the European sparrow? Is there something about that? I mean, does it have to be a generalist to actually infect a European sparrow?
That's another great question. I would have to. I don't remember offhand the parasite, the story exactly for those Passer domesticus. I think starlings got specialists. I think that there's some. I mean, I know that there's quite a few generalists that infect house sparrows. I just haven't looked at the data in a long time, so I'm not 100% sure. I think that starlings get specialists. yeah, it's also sort of unclear exactly what, how specialists evolve anyway. That was my alternative. That was going to be the alternative talk if I didn't talk about biogeography. And so that's also not clear. So yeah, I don't have a good answer to it. I think that it's I think that using some of these genomic techniques like sequence capture, there's transcriptomics and things like that, where we can get at other genes besides just the cytochrome B lineage, are going to open some doors and allow us to sort of understand better what's going on. I think we're on a kind of, it's going to be happening soon. Yeah, I think we'll have a better understanding. As far as the species delimitation, that's just yeah, fraught with controversy. You know, whether we call we in the avian malaria world, the species name, for instance, Hemiproteus nucleocondensis, it must be described from morphology of the parasite on the blood smear. And so, you know, two parasites that are diverging, and so as you can see with the GRW1, that Hemiproteus nucleocondensis, their barcoding region's the same. You have to go outside of it to see a very, very small divergence. They're probably on their, you know, they're isolated. You don't get great reed warblers hitting Seychelles, at least not regularly, not reported regularly. That's not part of their migratory route. And so, yeah, they're isolated. They're going to be diverging. How long will it take for that to reflect morphological differences? I don't know. Within the Plasmodium relictum clade, where you have that GRW4 that's in Hawaii, et cetera, these look very similar across lineages that are very diverged. And so, yeah, so it's a tough question. But so that I think the microscopy is super important, though, because it's how we can tell that a bird is a competent host. So we need to be able to link genetic lineages to the morphology of the parasite to be able to say, oh, there's that stage of the parasite in the blood of the bird, a gametocyte, as it were. That's the stage that when the vector picks it up, it's going to reproduce sexually in the vector. This is a competent host. Often, and many times, you screen a bunch of birds, you screen for malaria parasites, you just do the genetics, you find all these infections. But if you were to look in the microscope slides, you might not see all of the gametocytes. And that's undoubtedly because you have parasites that get into the blood of birds and they just cannot develop. They just can't, or they get a vector bites the bird, the parasite gets in, but it can't develop. It's a dead end host, at least for the time being. And so having that morphological data is just incredibly important to couple with the molecular data. The difference in the sort of how fast the morphology changes to be able to describe a species is just yeah, an unfortunate part of the biology.
It's the classical, we're trying to put a round peg in a square hole. I mean, so I think we all relate to that. Just that
actually, what you just said was really, it answered one of my interesting questions here, based on, you know, one of the slides that I've been presenting based on the meta transcriptomics, where the small subunit RNA was coming out, was that I had found in the same cohort of mosquitoes, I don't know if it was the same mosquito, but in the same trap full of Culex mosquitoes, there were two completely different lineages of Plasmodium. One was in the saliva and the other was in the excreta, which presumably would be things that weren't able to infect the mosquito and were just excreted. It seemed that the Valgani, if that's pronounced correctly, was in the excreta, and then the relictum was in the saliva. Which the reads completely segregated based on the collection method. The same small subunit locus, which I thought was really cool, and you just touched on it completely right there. So it seems like it's totally plausible.
You got relictum in your New Jersey mosquitoes.
Yeah.
Oh, that's great to know. Yeah, cool.
Yeah, it seems to be very prevalent. At least most of the places that we trap Culex, we also find Plasmodium.
Fantastic! Wow, cool. It'd be great to talk more about it.
Yeah, Francisco. So you really need to come and visit. Okay, Zoe, go.
Yeah, hi. I'm Zoe. I'm actually Dr. Price's graduate student, one of them. I had kind of a question sort of related to what he just said. I saw a paper from I think it was 12 years ago, saying that avian and malaria species aren't very closely co-evolved with their vectors. Is that still the thinking? I don't know too much about these parasites.
With their vectors.
Yeah, like if you do a phylogeny, is it really hard to like, you know, sort out that variable vector rather than a avian host?
I think there's a couple things that's an interesting point. There's a couple things to kind of consider. One is that a lot of the vectors are, not all, but a lot of vectors are generalist biters. And so you see sort of them, you know, a given vector species biting many kinds of bird hosts. And so then seems to be a lot of, you know, many vectors seem to be competent for many parasite species. And that sort of throws a wrench in the co-phylogenetic stuff. There's also many more parasites than the vector species. So yeah, you would need kind of tight clades of the parasites being, you know, associated with specific vectors in there. So I would doubt it. I'm not familiar with the paper, and I have never done that on my own. If I was just guessing, I would say probably not a lot of co-phylogeny signal there, co-phylogenetic signal. There's also not, there's very little phylogenetic signal with the vertebrate hosts as well.
Oh, right.
So, yeah, so that's also, that's been shown several times, and we've recently done that with actually with the Acrocephalus warblers and their Hemiproteus species. We use these full mitochondrial genomes to get better phylogenetic resolution on the Hemiproteus, and also no real signal. You find things like there's a group of parasites that infect vireos, for instance. A group of Hemiproteus parasites, all closely related, that infect vireos. And you see that kind of thing every so often, but yeah, but the co-phylogenetic, not as often.
But in some, I mean, it depends on what level. I mean, you have a group that infects culicoides and a different one that would infect black flies and a different one that infects mosquitoes. So it's just like, what level are you?
Absolutely.
Right?
Yeah, absolutely.
Okay.
Yeah. So when you zoom out and you look at the genus level, for sure, or the big clade level, absolutely. You have yeah, thanks for mentioning that, Dina.
Okay.
you have the Hemiproteus parasites of songbirds, which form a clade, are typically transmitted by biting midges. Many kinds of biting midges, but biting midges. But you have another group of Hemiproteus parasites that infects pigeons, the doves, the columbiforms, and also some seabirds like the frigate birds and things like that, that are transmitted by hippobocid flies, right? And you have Plasmodium transmitted by mosquitoes, and of course leukocytozoan transmitted by black flies. So yeah, at the bigger, that's a great point. At the deeper level of the phylogeny, yes, you have certainly specialized relationships between the parasites and the vectors. When you zoom in and get to the species level traits, harder, I think, would be to find that.
Yeah, but it's interesting to think about why would that be, right? Why would it be that you have, you know, it's just, I guess it's time, and which since I don't see any other hand up, I have to ask this question because you made a statement about Hawaii, and which is, you know, about the fact that there's these two PR8 and PR9, and you're sort of surprised that the I think it's PR9 is found in Kauai only.
Yeah.
But we don't, you know, when did it get there?
Right. Absolutely.
So it may not simply have gotten yet to the other islands because, you know, what are these samples for? What year were these samples collected, right? So this could be a new. Actually, what year were the samples collected?
Yeah, I'd have to check. I'm not sure. I think, but they're more recent. I mean, they're not that old.
Right.
They're not historical samples.
As you know, and I know Francisco has a lot to say about this, but you know, we're going to be meeting with you and others. But so there's this sort of hypothesis that there was a single GRW4 in Hawaii, and now clearly there's more evidence that there's multiple GRW4s in Hawaii. But then stating surprising that is only in Kauai could simply be because it got there 10 years ago, and you know, or last year or whatever it was, and just simply hasn't had the opportunity. Even the mosquitoes took some years to go across all the different islands. So, so
Absolutely, yeah.
so I also want to be respectful of your time. I know you need to teach. If so, it's noon. So,
Yeah, sure.
So are there any other questions? Again, we're going to probably Dr. Ellis will be coming over in person next Thursday, right?
Yes, yeah. Yep.
And so we'll send the schedule. We'll definitely have him here sequestered at CVB. So prepare to come over, but otherwise.
I'm not going to be here Thursday.
I'm
Oh,
sorry?
no.
I'm
Well,
at
okay,
AMCA.
he's gonna come back after Thursday, another day.
Oh, okay.
Because Francisco is and Julia are leaving, so sorry, Dana. He's coming to talk to Francisco.
He's not coming to see me?
It's not that far. It's not that far though. I think it's yeah, should be able to get up there.
It's exactly 2 hours, by the way. It's an hour and
Perfect.
45 minutes. So it's not a difficult trip.
Yeah.
So, okay, well, thank you. I'm just going to give a round of applause in vitro.
Yeah, thanks so much. Thank you very much for the invitation, Dina, and it was great to see all of you. And
Nice
yeah.
seeing you. And
Yeah, I look forward to seeing you in person.
All right, well, we'll see you soon.
Okay, thank you. Take care.
Bye-bye.
Bye.
Bye bye.
Bye.